Today, Aaron and I will be discussing our independent research project, which focused on the efficacy of varying concentrations of sodium hypochlorite on E. coli bacteria. We would like to first extend our greatest thanks to Dr. Andrew Eyre, who advised us in this process and gave us key insights into how we should be presenting this information today. Our project stemmed from our curiosity to learn more about disinfectants and their role in keeping surfaces clean during the pandemic. With an obvious understanding that we weren't going to be able to study COVID-19 specifically, nor all disinfectants, we decided to focus on the use of a widely popular disinfectant, bleach, and its interactions with a common bacterium, E. coli. We became interested in testing the most efficient way to employ bleach against E. coli because we recalled that in the early stages of this pandemic, many disinfectants were significantly limited in their availability. Consequently, we set out to test the efficacy of four different concentrations of sodium hypochlorite to ascertain the most efficient way to use sodium hypochlorite. After significant research, Aaron and I hypothesized that there would be a minimum sodium hypochlorite to water ratio or concentration that would work most effectively to kill E. coli. In our research, we found that two key compounds, which emerged when sodium hypochlorite reacts with water, as you can see below, hypochlorous acid and hypochlorite ions, were responsible for disrupting E. coli structure. Therefore, we hypothesized that there would be a threshold at which the sodium hypochlorite concentration would reach a minimum ratio to have just enough of hypochlorous acid and hypochlorite ions to cause E. coli to denature. Denaturation is the process of destroying a substance's characteristic properties, and in this case, it effectively means killing the E. coli. Based on information presented in Northeastern University's Environmental Health and Safety Report on commercial bleach, we predicted that the minimum sodium hypochlorite concentration would exist somewhere between 0.5 and 2% sodium hypochlorite. Although we could not find a specific study that focused on the relationship between sodium hypochlorite and E. coli, Annalena Severing and her colleagues did study the efficacy of commercial sodium hypochlorite on pathogens in 2018. Her study determined that there exists a concentration-dependent impact, finding that pathogens or bacteria showed significant decreases in activity at higher sodium hypochlorite concentrations. Severing and her colleagues worked with much higher sodium hypochlorite rate concentrations than we did, finding that the largest difference in efficacy for the sodium hypochlorite concentrations was between 10 and 75% against those various pathogens. Therefore, Severing's study showed that sodium hypochlorite has the ability to denature bacteria and that different concentrations have different efficacies against bacteria. Now I'm going to pass it on to Aaron, who's going to explain more about our methods during this process.
Yeah, as Quinn has already mentioned, we used E. coli in this experiment, considering it was a very easy organism to grow and study, as long as we followed fundamental safety protocols such as wearing gloves and goggles every time we handled the bacteria, and we had to disinfect our station after every workday. E. coli quickly reproduces under the optimal temperature of 37 degrees Celsius, and it's also convenient since we were able to subculture frequently throughout the experiment, meaning we could take small samples of E. coli and put them in new nutrient broth to use for future trials. For each trial, we started off by adding a few drops of bacteria into a container of pure nutrient broth. We then shook the container lightly and covered it up with tin foil and placed it in the incubator at 37 degrees Celsius for the bacteria to grow. Next, we had to take the bacteria culture out of the incubator and pour out 4 milliliters of it into a small cuvette. We then used a colorimeter, as you can see here on the bottom right, to measure the bacteria's absorbance at 635 nanometers of light before we treated it with the various bleach concentrations. The colorimeter basically passes a beam of light with a certain wavelength, in this case we did 635 nanometers, through a 4 milliliter cuvette and then calculates the percent of light absorbed by the solution. So if the calorimeter gave us a reading of 0.15, for example, that meant that 15% of the light was absorbed by the solution. We first had to calibrate the machine to a cuvette filled with only nutrient broth, after which we would be able to put in the new cuvette filled with nutrient broth and also bacteria to analyze the difference. One possible issue that might have occurred at this step in the experiment is the reliability of our colorimeter. At one point, the same sample of bacteria gave two different readings for absorbance at 635 nanometer light when we put it in at two different times. However, we do not believe this was a major concern as we repeatedly calibrated the colorimeter to pure nutrient broth solution after recording each measurement. Now we were ready to create the 4 different sodium hypochlorite concentrations. We did this by diluting the store-bought bleach, which was 5% sodium hypochlorite, with nutrient broth to each of the respective levels. After measuring the absorbance at 635 nanometer light, we transferred 3 milliliters of bacteria into 20 test tubes, which is resembled by the image at the top right. That's an example of the 3 milliliters of the E. coli bacteria. 5 of the 20 tubes would have 3 milliliters of bacteria and 1 milliliter of a 0% bleach concentration, which was really just pure nutrient broth. These tubes would be the control group in the experiment since we didn't actually manipulate them with any bleach. Another 5 tubes would contain 3 milliliters of bacteria mixed with 1 milliliter of the 1% bleach concentration. Another 5 would have 3 milliliters of bacteria with 1 milliliter of the 2.5% concentration. And the last 5 would have 3 milliliters of bacteria mixed with 1 milliliter of the pure 5% bleach concentration, which we just took straight from the store-bought bleach. Now we had 20 test tubes filled with 4 milliliters of bacteria and sodium hypochlorite mixture, which we would later use to measure the various absorbances and compare them to the previous calculation that we just took. We then took the 20 test tubes and put them back in the incubator to grow. And after a few days, we took the tubes back out and measured the new absorbance at 635 nanometers of light again. Time was the main challenge that we faced in this part of the experiment, since it was difficult to grow E. coli and measure the absorbance after 4 days, measure the absorbance of our samples on the same timeline. For example, in our first trial, we measured the absorbance after 4 days, as you can see in the bottom right here. But in the second trial, we were forced to measure the absorbance after 6 days. This inconsistency could have possibly contributed to some of the variability in our data. Either way, once we calculated the 20 absorbances, we placed the test tubes back into the incubator and recorded the data in a Google spreadsheet, as you can see here on the right. In order to analyze this data, we created an absorption ratio, which was calculated by taking the absorption after the addition of sodium hypochlorite and dividing it by the absorption before we added sodium hypochlorite. This let us compare data across all 4 trials, even though the absorbances for individual bacteria cultures varied. We successfully completed 4 trials with 5 cultures at each concentration of sodium hypochlorite, giving us 20 total measurements for each level. Our independent variable was the concentration of the sodium hypochlorite solutions. Our dependent variable was the difference between the absorbance level of 635 nanometers of light by the unexposed bacteria and the same bacteria following exposure to sodium hypochlorite for a certain amount of days, depending on which trial we were on.
All right, so now I'm going to summarize our findings. So this table here summarizes our overall trends pretty well, but it also highlights some of the conundrums and confusions that were presented in our results. Firstly, this first row here displays the various means we got, and they, as you can see, they go in a somewhat decreasing order from right to left. You'll notice that the 0% sodium hypochlorite solutions clearly has the highest mean absorption ratio, whereas the 5% clearly have the lowest mean absorption ratio. However, the 2.5% solutions actually had a higher mean absorption ratio than the 1%, implying that there is maybe no change in efficacy or even a decrease in efficacy between that 1 to 2.5% sodium hypochlorite range. This table also summarizes very well that we employed 20 different test subjects for each concentrations, but most perplexing from this is that you can see that the highest maximum value is this 1.651 value, which belongs to the 2.5% sodium hypochlorite solutions. Consequently, this could possibly bring in again into question the accuracy of our colorimeter, as Aaron had mentioned, as we saw some confusing results like this across the table. Now, this next figure here most clearly highlights the general trends in our data. This box and whisker plot clearly identifies the trend that a higher sodium hypochlorite solution will lead to a lower absorption ratio. For example, the third quartile for the 5% sodium hypochlorite solutions is lower than that of the first quartile for the 0% solutions, indicating that the 5% sodium hypochlorite solutions do indeed kill off some of the bacteria. And by that same token, this graph also shows that the first quartile for the 5% sodium hypochlorite solutions is at 0 on the absorption ratio metric, which indicates that 1/4 of our observations at the 5% mark killed all of the bacteria inside of them. Similarly, as we saw in the summary table, the 1% and 2.5% solutions don't really adhere to any trend at all. The third quartile for the 2.5% solutions is greater than that of the first of the 1%, whereas its first quartile is lower than that of the 1%. Consequently, this graph further implies the lack of significance we observed when increasing this concentration from 1 to 2.5%. Now, this last figure most clearly highlights the discrepancies and outliers that arose in our data. Though the means, these black dots, these black squares, again show the general trend that a higher concentration will lead to a lower absorption ratio. You can see at times how in, for example, trial 4, 5% sodium hypochlorite actually had the highest mean absorption ratio over that trial. However, if you follow each color of dots across the x-axis, they do generally follow the trend of the means, where 0% have the highest absorption, followed by 1%, followed by 2.5%, followed by 5%. Duly noted, however, is the fact that these mean absorption ratios for each trial have a range as great as 1 on the absorption ratio metric, which can likely again be attributed to the difference errors in the colorimeter or the differences among the different subcultures that we used for each trial. Now I'm going to pass it on to Aaron, who's going to summarize our findings for once and for all.
So to analyze our data, we performed a Wilcoxon signed rank test, which is a statistical test that allowed us to conclude whether the absorbance averages for the varying populations significantly differed from each other. From this test, we obtained 6 numbers known as p-values, which basically indicate if the difference between two populations is statistically significant or not. For this experiment, our significance level was 0.05, meaning that if the p-value was lower than 0.05, then there was a significant evidence of difference in efficacy between two sodium hypochlorite concentrations. Our results imply that there was a difference between the 0% concentration and the remaining concentrations that we tested, which means that even just a little bit of bleach is better than no bleach at all when fighting against E. coli. They also show that there was a significant difference with the 5% concentration compared to the rest of the levels, which proves that the 5% concentration was definitely the most effective against the E. coli bacteria. Surprisingly, however, with such a high p-value of 0.71, as you can see right here in the middle, there was no difference between the 1% concentration and the 2.5% concentration. So we cannot conclude that the 2.5% is in any way more effective than 1%. Based on these results, Quinn and I would be interested in focusing on a smaller range of concentrations between 0% and 5%, since 0% was definitely least effective and 5% was clearly the most effective. A future study could experiment with levels such as 1%, 1.5%, and 2%, and so forth to see whether there is any more accurate or more detailed threshold that our study might have missed. Another possibility would be to use a different bacteria in a future experiment, or even a different disinfectant, to see if the same methods that we use would result in similar data or different data. We specifically focused on how varying concentrations of sodium hypochlorite affect E. coli bacteria. So we cannot extrapolate and say that other disinfectants would have the same effect. But our experiment does allow us to conclude that 5% sodium hypochlorite has a significant impact on E. coli bacteria. But further studies would have to be done to reveal the effect that other disinfectants have on bacteria and viruses, such as COVID-19.